Hi everyone, my name is Ranah Ocampo.
My name is Summer Gibson, and we research the application of fruit flies in studying human neurodegenerative diseases.
Scientists have questioned how mutations globally expressed in proteins lead to neuron cell type degeneration. Huntington's disease is an example. It is an adult-onset dominant heritable disorder characterized by progressive psychiatric disruption, cognitive deficits, and loss of motor coordination. To further understand the pathophysiology of peripheral nerve and neurodegenerative diseases and how different mutations within the same gene cause them, we use the fruit fly as the model organism to investigate the molecules and mechanisms regulating axonal transport. We chose the fruit fly since it has been used in fundamental biomedical research for decades because of its anatomical simplicity. We focused on fruit fly gene P150 glued, encoding a protein vital in axonal transport. This gene is homologous to human gene dynactin subunit 1, which encodes a protein complex involved in diverse cellular function, which include bidirectional intracellular transport by binding to two motor proteins, dynein and kinesin 2, and linking them to organelles or vesicles to be transported.
Step 1 was finding the amino acid sequences for humans plus our 5 model organisms using NCBI's GenBank. Our model organisms are nematodes, fruit flies, zebrafish, mice, and chimpanzees. Once we had all 6 sequences, step 2 was using +Alt omega to align them. Step 3 was evaluating every position and determining the conservation among the organisms. In step 4, we boxed in the cytoskeleton-associated protein glycine-rich domain, also known as the CAPGLY domain, which we found through using ProCyte. Using UniPro, we found amino acid positions of natural variants within the CAPGLY domain that are responsible for 2 very different neurodegenerative diseases.
Third instar larvae were collected for the dissection. Third instar larvae is the life stage right before their pupation. Then we followed our dissection procedure by dissecting them in 1x phosphate buffered saline and 4% paraformaldehyde and PBS, and by pinning them down at the mouth and at the tail. We then made incisions at the top, bottom, and down the middle. After completing our procedure, we put the larvae in blocking solution and antibody to prepare them for imaging. The antibody used was fluorescent Affini Pure Go anti-horseradish peroxidase used in a 1:50 ratio. The antibody was used to bind to the neuronal membrane. Afterwards, images were required using a fluorescence microscope, and pictures were taken using an iPhone to locate the axons to determine if binding had occurred to indicate the accumulation of HRP punctae and to observe if axonal transport had been disrupted in the mutant flies.
At the bottom of our poster, you can see the amino acid sequences along with our color coding system. Yellow represents the sequences with 100% conservation, and blue represents those with more than 50% conservation. The black box represents the CAPGLY domain, which is positions 29 through 94. Below, we blew up a portion of the domain where you can see 2 other color markers. The red color represents the amino acid residue for HMN7B. The disease is similar to amyotrophic lateral sclerosis, or ALS. The magenta color represents the residue for Perry syndrome, which has mutations at 3 different positions. Perry syndrome is similar to Parkinson's disease. A major finding in the bioinformatics portion of our research is that the variant for HMN7B at position 59 and the variant for Perry syndrome at position 71 are extremely close to each other, but are very different diseases. In the pictures to the right, you can see the difference between the segmental nerves of wild-type and loss-of-function flies. In picture A, the nerves are labeled with anti-HRP, and we see homogeneous staining in the nerves. In picture B, with the mutant flies, we see uneven staining, indicating small swellings in the axons where the anti-HRP was clumped. This indicates an overexposure of anti-HRP.
We found that dynactin is conserved across multiple species. We also observed many natural variants within the CAPGLY domain that play an important role in neurological pathogenesis. Specific mutations 12 residues apart cause 2 distinct non-overlapping autosomal dominant neurodegenerative syndromes in humans. Moreover, an accumulation of HRP punctae along segmental nerves of the P150 glue loss-of-function mutant flies were observed. It is likely that the mutation would eventually cause axonal degradation due to the accumulations and traffic jams of cargoes on the neuronal highways.
In future work, we can study how axonal transport affects other organelles such as lysosomes and mitochondria. We can use immunofluorescence microscopy in studies like these as well to visualize these components. Thanks for spending your time watching our presentation, and I hope you find our work interesting.